Hello everyone, a very warm welcome to you all on the today's topic Wilson's disease. Here we will see the pathophysiology of the Wilson disease. Along with that, uh, we'll see the clinical features as well as morphology of the Wilson disease. Let's begin with the introduction. Wilson disease is a autosomal recessive disorder, and this mainly results from the loss of function mutations in the ATP7B gene. ATP7B gene it encodes a transmembrane copper transporting ATPase, which is present on the hepatocyte canalicular membrane. And loss of function mutation of this gene leads to impaired copper excretion into bile and failure to incorporate copper into the ceruloplasmin. And what does it do? It results in the accumulation of the toxic levels of the copper in many tissues. That could be liver, or it could be brain, as well as it could be eye. Next, we see the pathogenesis of Wilson disease. Here we see the source of copper is our food as well as water. Water contain various minerals within it, and these are the two source of the copper, where this copper will uh, be transported through the copper transporting receptor, which is present on the enterocyte, and this enterocyte helps in the absorption of the free copper from the GI tract, and later on this free copper will be transported to the portal circulation, which reach to the liver, and this happens with the help of the ATP7A protein channel. And next we see the portal circulation. Through the portal circulation, when the copper enters into the hepatocyte, then it goes through the this copper transporter receptor, which is present on the hepatocyte. um, it then enter into the hepatocytes, and that will go two pathway. Into the two pathway, one is excretion through the bile, and next it get incorporated into the ceruloplasmin, and this happen with the help of this ATP7B gene. And in this Wilson disease, what happens that there is a loss of function of this ATP7 beta B gene, and if this happens, then this both pathway will be impaired, and due to impairment of these two pathways, what happens that this free copper, which has been excreted through the bile, and 90 to 95% was being incorporated into the ceruloplasmin, that get impaired, and this free copper will be remained accumulated within the hepatocytes, and we can see this free copper taken up by the hepatocyte and incorporated into the apoceruloplasmin. This apoceruloplasmin is present in this Golgi bodies, where the ATP7B protein channel is present, and this free copper will be incorporated into that. And next, there is a deficiency of the ATP7B. Why there is deficiency? Because there is a loss of function mutation of the ATP7B gene, and that lead to the decreased copper transport into the bile. Here we can see if there is not a functional ATP7B protein channel, then the excretion of the copper through the bile will be impaired. And next, there is the impairment of the incorporation of the copper into the ceruloplasmin, that can be seen here. And next we see that the total serum copper may be lower than the normal due to the ceruloplasmin deficiency. What happens that once the copper get incorporated into the ceruloplasmin, then it will goes to the circulation, blood circulation, goes to the blood circulation, and its various activities will be performed later on with this. But there is deficiency of the ceruloplasmin. Why there is deficiency? Because there is the mutation of ATP7B gene, and due to this mutation, there will be the decreased formation of the ceruloplasmin, because the incorporation of the free copper into the apoceruloplasmin is impaired. So this happens, these two things happen in the loss of mutation of this ATP7B gene. Next we see the toxic effect of the copper to hepatocyte. We know that the free copper which get entered into the hepatocyte need to be excreted either through the bile, or that should be incorporated into the ceruloplasmin for it various activities. But these both pathways are impaired due to the mutation in the ATP7B gene. So the level of the copper in the hepatocyte will be more, and that will lead to various hazardous effect to the hepatocyte as well as other extrahepatic tissues. How it affect the liver as well as other extrahepatic tissues we discuss here. First is that the accumulated toxic level of copper lead to the formation of the radicals, free radicals, by the Fenton reaction. Here we can see, the every cell has a peroxisome, which lead to the formation of the peroxide, and later on these peroxide which are formed in the process of various metabolism, that gets reduced to water and oxygen, and that will be beneficial in case where there is the like NADPH, like molecules glutathiones. But when there is the toxicity of the copper, then it lead to the alteration of this S2O2 or other free radicals, and lead to the formation of the free radical, that is hydroxyl ion, and this is now called as Fenton reaction. And this Fenton reaction is facilitated by the Kuipers form of the copper, which has been accumulated into the hepatocyte, and this hydroxyl formation lead to the uh, destruction of the biomolecules, biomembranes, and various other effect. And most dangerous is that it may lead to the mutation of the genome, which later on may cause cancer. And this is how it lead to the hazardous effect to our body organs. Next, what it does is the copper bind to the sulfahydryl groups of the cellular proteins, and also displaces the metals that is present in the center of the metalloenzymes. And these two things that the excess copper when present does lead to the hepatic injury. And whenever there is the hepatic injury due to the copper toxicity, then the non-ceruloplasmin. Why non-ceruloplasmin? Because we know that in Wilson disease there is reduced formation of the ceruloplasmin. So non-ceruloplasmin bound copper is present abundantly inside the hepatocyte, and once there is injury to the hepatocytes, then those non-ceruloplasmin bound copper get spill over the uh, blood stream, and that will get circulated to other extrahepatic tissues, and that will lead to the accumulation of that non-ceruloplasmin bound copper into the certain tissues like the brain, basal ganglia, as well as eye. And next we see morphology. Morphology of the Wilson disease. Here we see there is the fat change or steatosis with a focal site necrosis. Why there is necrosis? Because we know that the copper toxicity leads to the Fenton reaction, which uh, often lead to the formation of the hydroxyl ion, which is free radical, and that will damage the tissues and lead to the necrosis. Next, there is the acute liver failure due to the damage to the hepatocyte, and uh, we know that after the injury to the hepatocyte, there is a spilling over of the copper accumulated in the hepatocyte to the blood stream, which later on get deposited to the extrahepatic tissues like eyes. And in the eyes, the deposition of the copper lead to the formation of the Kessler-Fleischer's ring. And next, we can see in some cases of Wilson disease, there is a chronic hepatitis, and this chronic hepatitis is characterized by the admixed fatty changes. Why admixed fatty changes? Because of the free radicals that has been produced due to the Fenton reaction that has been induced by the toxic level of the copper into the hepatocyte. And next, there is the moderate to severe portal inflammation as well as the hepatocyte necrosis, which is similar to this one, but next, what extra thing happen in this chronic hepatitis in case of the Wilson disease is that there is the feature of steatohepatitis. What features are there? There is the ballooning of the hepatocyte, and there is a prominent mallory haline and perisinusoidal fibrosis. These things happens during the chronic hepatitis. And next, we see the clinical features of the Wilson disease. Here we can see after the hepatic injury, the spilling over of the copper lead to the accumulation of the copper into the various extrahepatic tissues, and this accumulation lead to the toxicity of those tissues. For examples, if that get accumulated in the brain or in the basal ganglia, then it will lead to the manifestation of the various psychiatric disorders like movement disorders, difficulty in speaking, as well as cognitive impairment, and psychiatric disturbances like the psychosis, mood and personality disorders. And even if there is the accumulation of the copper in the eye, in the cornea of the eye, then there will be the formation of the Kessler's Fleischer's ring. Here we can see there is a brown colored ring which is due to the copper deposition. This is called as the Kessler-Fleischer's ring. And next, that in the liver there is a steatosis, that is the fatty changes in the liver, fat accumulation in the liver. Metabolism is affected since hepatocyte has been exposed to the free radicals, so the metabolic activities inside the hepatocyte has been impaired, and that will lead to the inflammation as well. And later on in the process of the healing, or in the process of you know, the healing of the wound, there is the fibrosis of the hepatocytes, which often lead to the cirrhosis. And next, we can see through the blood circulation, the copper has reached to our joint, where it may lead to the osteoarthritis, osteoporosis. Why? There is the accumulation of this copper, and this will, everywhere it will lead to the Fenton reaction, and this will produce free radicals, and later on again that will lead to the reactive bone formation, or there is the uh, degeneration of the osteoblast or osteoblastic cells, and also various inflammatory responses will come to occur against this copper tox- toxicity. Next, we see that this copper is producing free radicals, which is harmful to the RBC since its oxidative stress will increase. And in case of the oxidative stress, the membrane of the RBC will be damaged, and This destruction of the RBC membrane led to the hemolytic anemia due to the lysis of the RBC. Thus, in case of copper toxicity, we can see hemolytic anemia as well, and we know that the uh, copper is present excessively in the bloodstream due to the spilling over of the copper from the liver to the various um, blood circulation, and that will lead to the increased urinary copper excretion and the renal tubular dysfunction. What happens that the